Hello, my name is Josh Salet. This is a pre-recorded presentation for the 2020 meeting of the Society for Mathematical Psychology. I will present a theory of temporal preparation, the cognitive function that takes place when anticipating a future event. Here, I present a computational model, fMTP, that describes preparatory behavior across different timescales. The message that I'm going to convey in this presentation is as follows. First, reaction time phenomena of temporal preparation showed to be modulated across a range of timescales. Second, thus far, hazard-based models fail to account for all phenomena. Third, we therefore present an alternative model, fMTP, in which we integrate ideas on motor planning, the coding of time, and episodic memory. Finally, I will show that fMTP accounts for all phenomena observed in temporal preparation. First, I will cover the observed empirical phenomena. Since the early 1900s, cognitive scientists have studied temporal preparation using the classical four-period paradigm. In this paradigm, a cue, the fixation dot in this example, indicates that after some time the target will appear, to which participants try to respond as fast as possible, which is in this case indicating at what side the square is presented. The interval between the cue and target is known as the for period. The observation that responses to the target are faster when cued gave birth to the study of temporal preparation. This process has been widely studied by varying the for period. The typical finding is that reaction time, here plotted on the y-axis, decreases as a function of for period, plotted on the x-axis, reflecting the development of temporal preparation throughout the for period. The more time left before the target appears, the more time there is to prepare, and therefore responses are usually faster. Over the years, this reaction time for-period function has been extensively studied, revealing that preparatory behavior is modulated across a vast range of timescales. In the next few slides, I will shortly overview these effects. On a shorter timescale, temporal preparation is modulated by the immediate preceding trial. Again, we see reaction time plotted on the y-axis and for period on the x-axis. But now, additionally, reaction time will be plotted as a function of the previous for period. This reveals a rather complex pattern. In short, the key here is that in the case of a previous long for period, responses slow down for the short current for period. And this effect vanishes as a function of current for period. On the intermediate timescale, the average reaction time across trials within an experimental block depends on the distribution used to draw the for periods from. Typically, for periods are uniformly distributed. That is, each for period is equally often presented within a block. As we saw previously, in this case, reaction time decreases as a function of for period. Alternatively, one could use a higher proportion of long for periods within a block, known as the anti-exponential distribution. In this case, reaction time for-period function becomes more expressed. In the case of a distribution containing a higher proportion of short for periods, known as the exponential distribution, the reaction time for-period function becomes flat. On the longest time scale, temporal preparation is modulated across experimental blocks. This was shown in an experiment that was divided in 3 different phases with 2 groups of participants, the anti-exponential and exponential group. In the pre-acquisition phase, both groups received the same uniform distribution, as displayed on the left bottom. As expected, the reaction time for-period function for both groups were similar. In the subsequent acquisition phase, the exponential group completed blocks with a higher proportion of short for periods, as displayed in orange, whereas the anti-exponential group completed blocks with a higher proportion of long for periods, as displayed in green. This resulted in a typical diverging distribution modulation for each group. No surprises so far. Crucially, in the test phase, both groups receive again the exact same uniform distribution. The reaction time for-period function revealed a transfer effect. Although both groups completed blocks with the same distribution, the reaction time for-period functions differed between the groups. This difference reflected the shape of the functions from the previous acquisition phase. These effects across a vast range of timescales pose a challenge to theorists. What is the unifying cognitive mechanism giving rise to this family of temporal preparation effects? One of the most popular accounts in the literature is the hazard function. However, such theories fail to account for all phenomena. In the next few slides, I will shortly discuss the hazard function. The hazard account of temporal preparation comes from the observation that the reaction time foreperiod function is correlated with the hazard, the conditional probability that an event will occur. As one can see, for the uniform distribution, the hazard increases as a function of foreperiod. As more time is passing, the more likely it is that the target will appear soon. The hazard account earns its popularity as flipping this upward sloping hazard gives rise to the downward sloping reaction time foreperiod function. Additionally, the hazard function develops differently for different distributions. As displayed in the middle panel, it steepens for the anti-exponential functions and flattens for the exponential functions. Again, flipping the hazard function seems to be correlated with empirical reaction time foreperiod functions of these distributions, as displayed in the right panel. However, the hazard account falls short when observing the reaction time foreperiod function across different timescales. It simply cannot explain effect of previous trials nor effects from previous experimental blocks. Furthermore, although the hazard functions seem to be correlated with the empirical data, mapping the hazard directly on reaction time, as displayed with the dashed lines in the right panel, reveals that there is not such an obvious relationship between empirical data and the hazard rates. Finally, although the hazard function might roughly describe the distribution effects mathematically, it remains ambiguous about the underlying mechanism. It remains unclear if and how the brain computes the conditional probability of the distribution density function. Given the inequality of the hazard, we present an alternative model, fMTP, Formal Multiple Trace Theory of Temporal Preparation, in which we integrate ideas of motor preparation, interval timing, and episodic memory. I will first go into fMTP's motor and timing components. fMTP assumes the preparation results from an interplay of motor inhibition and activation. In anticipating of the target, motor inhibition, displayed here in red, is applied to prevent premature responses. However, at target presentation, as 2 here, inhibition is suppressed and the response representation is activated, as displayed in green. These are the basic assumptions that are supported by a large body of neurophysiological evidence. fMTP states that this interplay between inhibition and activation is not a static process, but a dynamic process developing throughout the foreperiod. Therefore, the inhibition and activation process is supported by a time representation. To integrate such a representation in fMTP, we borrow a computational framework from Schenker and Howard that actively builds from the properties of so-called time cells. These cells represent specific time points in the foreperiod. The heat map, displayed here on the left, represents a layer of these cells in our model. As displayed, some cells, like T1, show to represent early time points in the foreperiod, where other time cells, like T3, show to represent later time points in the foreperiod. As displayed in the middle, we couple the time representation to the inhibition and activation units. Key to fMTP is that each of the time cells forms Hebbian associations with those motor units, reflecting fMTP's memory mechanism. The strength of the Hebbian association is determined by whether the time cell's activity, displayed now in the bottom of the right panel, co-occurs predominantly with inhibition or activation. As depicted in the bottom right panel, the activity of early time cells, like T1, predominantly co-occur with the inhibition node, and therefore make strong associations with this unit. In contrast, later time cells, like T3, co-occur predominantly with activation, and make stronger associations with the activation node depicted by the green surfaces. After an example trial, the Hebbian weights between the time cells and the MOTI unit, displayed here in the middle of the figure, are updated accordingly. This is visualized on the next slide by a change in the thickness of the connection lines. The effect of these weights on the dynamics of the MOTI units, now depicted as two block signals in the right panel, are illustrated on the next slide. As displayed, the block signals are adjusted such that they reflect the dynamics of the previous 4 periods. The critical assumption of fMTP is that temporal preparation is driven by the memory of all previous timing experiences, collectively determining the preparatory state on future trials. How does that work? Here I displayed the history of 6 trials of 3 different 4 periods. These traces will determine the preparatory state on the upcoming trial. However, each trace contributes according to its recency. Due to forgetting, older experiences, displayed on the top, are less readily accessible and thus contribute less strongly to preparation compared to the more recent experiences that are displayed in the bottom. To arrive at a measure of preparation, we first summed all previous traces according to their recency and took the ratio between activation and inhibition. In the next animation, I visualize this process. In the left panel, you will see the development of the preparatory state of the network, and in the right panel, you will see its effect on reaction time. Thus, as a consequence of its recency weighting, fMTP contains short-term dynamics contingent on the previous 4 period, displayed in the left panel, and at the same time, as a consequence of the everlasting contribution of each memory trace, fMTP contains long-term dynamic contingents on the 4 period distribution, as displayed on the right panel. In this part, we will contrast fMTP to the typical temporal preparation effects across the timescales. To do so, we simply translated fMTP's preparatory states linearly to reaction time. I would like to stress here that the same set of parameters is used for all empirical effects I will overview here. The canonical 4-period effect in the temporal preparation literature is the downward sloping reaction time 4-period function for the uniform distribution. Indeed, displayed with a dashed line, fMTP closely replicates this finding. On the shortest timescale, temporal preparation is modulated by the immediate preceding 4 period. Again, fMTP closely replicates this pattern for each of the preceding 4 periods displayed one by one. The only data point not captured by this fit is the first blue data point on the left. However, in most datasets, this empirical data point has a similar value as the other blue data points, resulting in a flat line from a previous 4 period of 400 milliseconds, as is predicted by fMTP. On the intermediate timescale, we saw the effects of the 4-period distribution on temporal preparation. Again, we can appreciate fMTP's close fit for the uniform distribution, anti-exponential distribution, and exponential distribution. On the longest timescale, transfer effects emerge, reflecting the contribution of previous experimental blocks. For this effect, we concentrate on the test block, depicted on the right, revealing the transfer effects. As depicted on the right, fMTP also replicates such long-term effects. To summarize, the message that I conveyed in this presentation is that a family of temporal preparation effects emerge across a range of timescales. Hazard-based models do not account for this family of effects. We present the formal multiple trace theory of temporal preparation and show, by integrating insights from research on motor planning, time perception, and memory, to capture all reaction time phenomena. I would like to thank you for listening and would like to thank my collaborators Wouter Kleine, Sander Los, Hendrik van Rijn, and Martijn Meter.